Hello, everyone. Welcome to the fall 2023 edition of the Include Data Coordinating Center webinar. My name is Joaquin Espinosa. I serve as the leader of the Administrative and Outreach Core for the Include Data Coordinating Center, who is hosting this webinar today. And good morning to some of you. I can tell that for some of you, it's good afternoon. And I can see some colleagues for whom it's even good evening. So feel free to introduce yourselves here in the chat. If you want to put your name, your institution, your locality. Part of the reason to do these webinars is to create community among Down syndrome researchers and others interested in using the Include Data Hub. So I'm going to go ahead and model this. I'm going to put my name here. University of Colorado. There you go. Feel free to introduce yourself. So our first presentation today will be from Dr. Huiqing Li from the National Heart, Lung, and Blood Institute. She's the project scientist for the Include Data Coordinating Center, meaning she's one of the many NIH officers who helps develop and run the Include Data Coordinating Center and the Include Data Hub. Huiqing, all yours.
Thank you, Joaquin. Let me share my screen with you. Okay, can you see my screen well?
Yes, looking very well.
Great. Good afternoon, everybody. Welcome to the 2023 Winter Include Data Hub webinar. Thank you, Joaquin, for your introduction. My name is Huiqing Li. I'm a program director at the National Heart, Lung, and Blood Institute. I'm currently serving as the project scientist for the Include DCC. So today I want to share with you some information opportunities from the Include project. As you may know, that Include project launched in 2018 by a congressional directive. And from 2011 to 2024, you can see the funding, the NIH funding on Down syndrome. And for Include awards from fiscal year 2018 to 2023, the total accumulative funding is 348 million. And in fiscal year 23 only, the NIH funded an estimate of total 130 million in Down syndrome research, and among them, 90 million were awarded to the Include projects. As you may know, that Include project has three components. Component 1 focuses on basic science. Component 2 focuses on cohort development. And component 3 focuses on clinical trials. And the Include Data Coordinating Center is part of the component 2. The mission for the Include Data Coordinating Center is to create an accessible data resources and a global Down syndrome knowledge ecosystem, which is the Include Data Hub. I hope you could appreciate the benefits of data sharing, which can fostering transparency and validation of research results, so that it can increase by increasing statistical power and empowering new scientific questions and innovation, which I think is very important for rare disease and pediatric studies in general. Eventually, data sharing can facilitate the development of and improvement of therapeutic and diagnostic strategies for patients. So if you are interested to apply for Include funding or other NIH-related Down syndrome related funding opportunities, we highly encourage you to list Include Data Hub as your data repository in your data management and sharing plan. If you are ready to share the data through the Include Data Hub, we will ask you to fill in an intake sheet. And for the next step is for the Include Data Coordinating Center have a listening session with you to learn more details about your data. If you have any questions, please feel free to reach out to the DCC team at info@includedcc.org or to myself. We also have data sharing FAQs on the Include website, and you can also see the current funding opportunities on the website. So the latest novels we published is the EXO-1 PAR, which was published 2 days ago, and I believe you haven't received the email from NIH Guide already, and you haven't received the email from NIH Guide yet. So this is probably the first time for you to know these funding opportunities. I will share my slides with you after the talk, so you can click to the link to get the information. The other opportunities the Include Data Hub is providing is called the Cloud Credit Pilot Program. So the Include Data Hub has not only a portal, but also a cloud-based platform, Kvatica, so where researchers can run bioinformatics workflows and interactive analysis using R or Jupyter Notebook. So the Include project is allocating cloud credit for users on Kvatica. You can learn more from this link or just scan the QR code. And to give you more details about the Cloud Pilot Program, there are different tiers of cloud credit distribution. The first one is $100 pilot price to all Include users. And the second tier is 1,000 limited cloud credits to if you have a brief description of your product provided, and if you're using Down syndrome data or co-analyze with Include Down syndrome data. Once you finish your 1,000 cloud credit, you can, if you need additional cloud credit, you can submit a detailed description of your future use of the cloud credit, and we'll have an administrative review so that after approval, you can get up to 20,000 extended cloud credit. So currently, we have 10 cloud computing products on Kvatica. So that's all I have to share with you. Let me know if you have any questions. Thank you.
Thank you very much, Hui-Ching, for the introduction. And for those of you who signed in a few minutes late, we've been doing introductions here in the chat, name, last name, institution, location. I'm happy to see we have colleagues from Latin America, from Asia, from Europe, a very international, very cosmopolitan group of people today. So I'm going to move into my presentation. I apologize in advance to those of you who already heard my introduction. Hopefully there'll be some new content here relative to other webinars that we've done in the past. Again, my name is Joaquin Espinosa. I'm the leader of the Administrative and Outreach Core of the Include Data Coordinating Center. And as Hui-Ching well said, our mission is to accelerate discoveries that improve and enrich the lives of all people with Down syndrome through accessible data resources that enable collaboration, connected communities, and the expansion of a global Down syndrome knowledge ecosystem. Now, in 2019, the NIH called for the formation of a data coordinating center to accelerate the discovery on the mechanism underlying the co-occurring conditions of Down syndrome by facilitating access to and querying of data from studies of people with Down syndrome. And NIH called for this data coordinating center to be organized in 3 cores listed here: the Administrative and Outreach Core to provide program management, outreach, and education; the Data Management Core, managing all the data collection, processing, and harmonization; and the Data Portal Core to facilitate access and analysis of the data via an online web portal, which we now call the Include Data Hub. So this is our team. It's a large team, very diverse, very multidisciplinary, 7 principal investigators listed here. I lead the administrative and outreach core out of the Cernic Institute in Colorado. Then we have Brian, Melissa, and Robert running the data management core, and then Adam, Vincent, and Jack running the data portal core. And this is a cooperative agreement with NIH. Huichin is our project scientist, but there are many other NIH officers, not only on NHLBI, but also at other institutes who help us with this important work. Now, if you understand the acronym of the Include Project, you know that we are all about the co-occurring conditions of Down syndrome. This is the first time in history that we have a very sizable population of individuals with Down syndrome that are living into late adult years. And now we know that they have a different disease spectrum or a different clinical profile, as we like to say. So on the good side, they are protected from many co-occurring conditions that affect the general population. It's very rare to hear about, say, an adult woman with Down syndrome having breast cancer. It's very rare to hear of adults with Down syndrome having atherosclerosis or hypertension. Now, unfortunately, they are highly predisposed to a number of co-occurring conditions that are decreasing their quality of life and also shortening their lifespan. So if we are to serve people with Down syndrome, we must study the mechanisms by which this extra copy of that small chromosome, chromosome 21, is creating these risks for various co-occurring conditions. Just to give you some numbers, and I'm mindful that these numbers apply here to the USA. They may be different in other parts of the world. A baby born with Down syndrome in the USA today has a 50% chance of having a congenital heart defect, 10 to 20% chances of developing a dual diagnosis of autism spectrum disorder, much increased risk of complications from lung infections, 60% or more chances of developing autoimmune conditions, and a mean age of Alzheimer's onset, which is much earlier than in the general population, 53 years old, and a mean age of death, 58 years old. So clearly, we have a lot of work to do here with Include Data Coordinating Center to help people with Down syndrome elongate their lifespans and enhance their well-being. So how are we going to do this? We're going to learn from people with Down syndrome. People with Down syndrome, they all have this extra copy of chromosome 21, but they are dealing with that trisomy in a very personalized, very unique way. And it's in that diversity, that heterogeneity, that we believe is an opportunity for discoveries. How come some of them have some of these co-occurring conditions, but others don't? Some of them may have a very early onset of Alzheimer's disease, others don't. Even some of the core phenotypes, like stunted growth, for example, or neurodevelopmental delays, can be highly variable in this population. So we clearly need to develop a precision, personalized medicine approach for the study of Down syndrome. So what is driving this heterogeneity? It could be influenced by genetic variants, due to the ancestral lineage of different people with Down syndrome, or it could be variants on chromosome 21 genes or on modifying genes elsewhere in the genome. Could be due to epigenetic mechanisms, not changes in the DNA, but things that happen later during development. Could be that these different phenotypes are matched by different endotypes, different variations in the transcript of the protein, the metabolome, the microbiome, and so forth. And of course, there could be roles for socioeconomic status, rate, and ethnicity, lifestyle, cultural factors, diet, so on and so forth. So clearly, there are a lot of ideas, a lot of hypotheses. We need to have an orchestrated effort to be able to accelerate all this increase. So I'm very happy to say that the team worked very, very hard since we received the funding in late 2020. And in a year later, we had the DCC website with a lot of important information. And on World Down Syndrome Day 2022, on March 21st of 2022, we had the first version, the first iteration of the Include Data Hub. So these efforts are clearly driving a burgeoning ecosystem of data resources for people with Down syndrome, researchers, families. The first research was the DS Connect registry run by NIH, which continues to be a great source of information for self-reported data from advocates and caregivers, but also it's a great recruitment tool to studies. Also many important community resources. Later, we had the Trisom Explorer, the online portal of the Human Trisom Project. But now, thanks to the DCC, we have this website with an investigator registry, publications and grants, a help center, a newsroom, and as you will see today, an Include Data Hub with a lot of cool features, very large-scale data sets, many studies, harmonized data, cloud computing, and access to both register and control access data. And we're working very hard to serve also what Huichin called the component one investigators, those that are doing basic science studies using animal models, iPSCs, and other systems. We're developing a portal for them as well that we hope to tell you more about in the near future. So if you create an account in the Data Hub right now, look at this. You'll see 11 studies that have already provided data on more than 9,000 research participants, more than 7,000 of them with trisomy 21, and here are the logos of the various studies that have participated and the various numbers. We'll take any data, we'll take any number of participants. No study of Down syndrome is too small for us or has less importance. So if you want to contribute data, please reach out to us. So you go there into the Data Hub, look at this. This is amazing. 9,000 harmonized clinical profiles, more than 4,000 whole genome sequences, more than 1,700 transcriptomes, proteins, metabolomes. And if you look at our pipeline, you will see we have epigenomes, immune maps, microbiomes, images. We have a lot of other cool data sets coming online soon. So, of course, we're doing this because we're trying to produce a cultural change with the guidance of NIH. We want to move away from data hoarding into an open science framework. So this is what's happening, or what has happened for the longest time. We all pay our taxes here in the USA. We do to this agency called the IRS, and then the Congress gives NIH, the largest funder of biomedical research in the world, a lot of money, billions of dollars, so that we researchers can apply for the funding and do science. And we spend most of that money generating data that most often than not ends up in an obscure place at our local universities or centers. And then now and then we slice that data, we publish a paper, oftentimes in a journal that has a very expensive subscription. So we like to say that that is not fair. The data is not findable, it's not accessible, it's not interoperable, it's not reusable. We want to shift to a paradigm where the first part of the equation is the same, but now we reward scientists, we reward each other by releasing our data in a way that is fair. And of course, we still like to write papers, but hopefully in open access journals. But everything is transparent and everything is accessible. The data is open, the code is open source, the methodology is very clear, so that people can reproduce. The peer review is transparent, the journals are open access, even the educational resources are accessible to everybody. So if we all manage to do this, if we embrace this new culture of open science, we'll be able to have a massive cohort of people with Down syndrome here in the Data Hub, leading to great sample sizes and increased statistical power, more diverse cohorts. We'll make sure that everyone is well represented in the study of Down syndrome. We'll be able to study conditions that are perhaps less common, where any one cohort may not have enough data, but if you combine multiple cohorts, now you have the data that you need. We'll be able to cross the pediatric adult divide. We'll have cohorts that are enriched for kiddos, others for adults. We'll be able to harmonize the data and look at the entire picture. We'll be able to assess reproducibility of findings by setting up discovery cohorts versus validation cohorts. We'll be able to explore the data in the hub before ordering a bunch of expensive reagents in the lab. As you will see today, you'll be able to identify available biospecimens and set up collaborations with those biobanks, and also collaborations with other like-minded scientists that may have a common interest. So I'm very happy to report that in the last 12 months, we saw a lot of activity, a lot of papers coming out of the first analysis of the data sets in the Data Hub. The Human Trisom Project team in December of last year published this paper reporting IGF-1 deficiency downstream of growth hormone signaling in Down syndrome. And that is associated not only with the stunted growth phenotype of people with Down syndrome, but also with elevated markers of neurodegeneration and neuroinflammation in the bloodstream. Shorter children with Down syndrome have a stronger IGF-1 deficiency, but also higher markers of neurodegeneration and increased risk of some co-occurring conditions. This is leading to a lot of activity, a lot of interest in the possibility of clinical trials for IGF-1 growth hormone in Down syndrome. Earlier this summer, we had another set of data sets analyzed by the Human Trisom Project. In this case, it was whole blood transcriptome and inflammatory markers in plasma, doing an analysis of chromosome 21 gene overexpression, showing this variegated overexpression. You'll hear a lot more about this from Micah Donovan next, how different people with Down syndrome express different sets of chromosome 21 genes. And then there's a lot of animal experiments here showing that triplication of interferon receptors encoding chromosome 21 in humans drives multiple hallmarks of Down syndrome in these animal models. Later in the summer, we had yet another paper, a lot more data sets released to this paper, also in the Data Hub, a multidimensional definition of the interferonopathy of Down syndrome, ranking people with Down syndrome from high interferon activity to low interferon activity, and seeing how that impacts on their transcript of the immune cell types, the co-occurring conditions, so on and so forth, and a case study of a participant with Down syndrome taking one of these JAK inhibitors to reduce the interferon hyperactivity to great therapeutic benefit. These two papers justify 2 ongoing clinical trials for JAK inhibition in Down syndrome. So a lot of exciting activity out of the data sets in the Data Hub, but this is something that I want to emphasize. The data sets in these papers were released in March of 2022. And look at the papers came later in December of 2022, June of 2023, July of 2023. And if you read the data availability statements of these manuscripts, you see that they are pointing to the Synapse platform and the Include Data Hub as the repositories for the data. So if you have data and you're writing your papers, do not wait until the paper is published. Go ahead and say in the paper that your data is already deposited in our repositories, right? So engage with us ahead of publication so that when your paper comes out, you can be proud that your data is open access through the Include Data Hub. Wrapping up here, I want to advertise something that we've been doing for a couple of years here, the Data Science for Diverse Scholars in Down Syndrome Research, or the DS3 course. It's a training event sponsored by the Include Project for diverse scholars who want to learn how to perform fair research in the Include Data Hub and other environments. We also train the students in the skills necessary for data management, analysis, and visualization, and also other skills required for career advancement. This past summer, we had the 2nd edition of the DS3. This is a very diverse, very colorful class, 24 students from all over the USA, even some from Europe, 9 instructors, 12 guest speakers, 2 weeks of very immersive training, a wonderful experience. We even had a yoga class with a self-advocate. You can see very hands-on instruction, but also, of course, lectures from invited speakers, presentations by the students themselves, and presentations by the instructors in the Include Data Hub. So we are very hopeful that we'll be able to repeat this next summer. So please mark this. You will, if you have subscribed to our newsletter, you will be getting notifications about this. We're holding these 2 weeks in July of next year, July 8th to the 19th, to have the 3rd edition of the DS3. And in the spring, you'll be seeing some action here in terms of applications and so on and so forth. So finally, please subscribe to our newsletter. If you go to includedcc.org, you will find this little button here, subscribe. Give us your email address so that we can keep you updated of new data sets, new webinars, new training events. And also, if you are an Include funding investigator, you should have a profile in our website. Here's Philip's profile with your grants and your publications. We're about to upload a new batch of Include funded investigators. So please make sure that we're representing everything correctly. This is a semi-automated population of this website. So if we get anything wrong, please feel free to let us know. And if you want to keep learning or improving your Spanglish, tomorrow I'm going to be doing a longer presentation about other things that I didn't cover today related to data sharing and reuse in the Include project. We will pass the Zoom podium to the leader of the Data Portal Core, Dr. Adam Resnick from Children's Hospital, Philadelphia. Adam, all yours.
Thanks so much, and it's a privilege to present on behalf of, as Joaquin, pointed to the introduction, a really diverse and multi-institutional team that comes together on behalf of building out these resources. my own background is in the neurosciences, and we've been at this process of developing portals and resources for communities performing research for some time. And increasingly, what Micah just presented is a recurring narrative, which is as you begin to try and leverage the multi-omic context of our continuously evolving capacity to generate very deep multi-layer data sets through new technologies across sample-based cohorts and intersect that with rich clinical phenotypes that span across time on behalf of a precision medicine setting, invariably, you end up coming up against the limit of what you can do as a single institution or a single laboratory. And in part, that defines the mandate under which the Include Data Hub continues to operate. And for those of you who have not been to other webinars, I thought I'd just go through a couple of different key elements of the Data Hub and then set the stage for additional developments and features that have been developed by the team as part of the Data Hub, especially following up with Perret and JP. As Joaquin mentioned, one context of acceleration is data sharing and rapid access to data, where access is not the limiting factor to the discovery setting across a multidisciplinary landscape of researchers. The other part that we aspire to is to accelerate through the use of technologies that supports this accelerated framework, support that acceleration through a collaborative framework of use of the data in the context of secure and governed cloud-based environments. Wei Ching mentioned the affiliation of the portal with a cloud-based environment that now has cloud credits to support it. And that this ecosystem needs to be fairly agile to continue to support the kind of multi-omic cohort that Micah just highlighted, where you not only create this heterogeneous mixture of data sets and files, but can accommodate the different communities that typically analyze and work on those data from their own domain of framework. The underlying methodology of the portal and data hub really is framed by this continuous flow of highly annotated, clinically annotated, or health record or participant annotated samples, where from those samples, large-scale multi-omic data sets are generated, like what Micah just highlighted. But those multi-omic data sets are complex and need to be harmonized and integrated across the multiple entities that are generating that data into information that then the portal and the platform supports as a queryable and discoverable framework. And that discoverability has to be connected to the use and enablement of that data by the community to convert that information into knowledge that ultimately drives impact. And this, to be transparent, is an ongoing iterative process that we continue to try to improve and continue to inform with new technologies as we move forward. To date, what we have really established is this middle conduit, this most critical part to go from the left to the right of this pipeline requires for us to have a portal where users can interact with the data files, the clinical features, and then access those files on behalf of their use within diverse environments. So users can intersect cohorts across clinical features, data file features, and you'll hear more about biosample features, and then create their bag of data of interest and then operate, analyze on that data within a supported cloud environment that's immediately actionable, and at the same time allow them to intersect their own data in an ongoing basis into this rich growing cohort of sample-based files and clinical features. Over the past year, we've also further empowered the integration of other data sources that might be relevant within the Include Data Hub to create an interoperable cloud environment. And so many of the co-occurring conditions within the Down syndrome setting have entirely separate cohorts in the non-Down syndrome context, like cardiac birth defects, for example, in the setting of the Kids First portal and cloud environment. And over the past year, we've been able to operationalize and link those resources in a very facile and seamless way that again allows the enhancement of the portfolio of data sets that can be analyzed in one environment across cloud features and within a secure and governed framework that manages the appropriate access and use of those data. One of the hardest part about operationalizing these frameworks is that while the digital domain of data creates fluidity, access, distribution, non-locality, there's always an analog to digital interface. That analog to digital interface that we have worked on to date has been the human trying to access data in the digital interface. But in all of these efforts, a key component is the analog to digital interface at the biosample layer. And empowering that is a continuous challenge for the entirety of the community and research landscape, not just in the setting of Down syndrome, but more broadly. There are many reasons for this. In part, these reasons span the unmet need of resources to collect and analyze samples for research as distinct from sample-based analysis that occur at the clinical interface. And then more historical reasons exist in the fact that these biosamples historically have been managed by biorepositories whose main and persistent focus is on managing the samples. And those biorepositories historically are by and large data disconnected. They operate largely on managing specimens, IDs, and distribution of those samples in ways that is disconnected from the data generation framework. And this is in part really just a vestigial feature of the fact that LIMS and biorepositories existed far in advance these large-scale multi-omic capacities of biosample-based resources. And so some of the work that you'll hear about next from some of our partners really focus on this setting and how can we develop new frameworks for interacting between this analog biosample collection, curation, and distribution features of our community and linking that into the ongoing rapid analytic framework that the portal empowers. We knew as a data resource core that we would have to address this from the very beginning. And so even from the very beginning and the launch of the portal, you might have noticed that biosamples and biospecimens were really first-class citizens within our portal. Most data portals do not necessarily focus on biosamples as a first-class entity, in part, again, for vestigial and historical reasons, because biosamples in that workflow is typically considered as a separate and non-connected framework for research. Excitingly for us, working with our colleagues and partners at NIH, we were able to initiate a virtual biorepository pilot of the past year that looks to challenge that model of disconnection across the analog and digital setting of biorepositories and ask the question of whether or not we can leverage these platforms that we're building in the Include Data Hub on behalf of an acceleration framework of ongoing and iterative research in ways that can allow a community to not only find data products and data assets generated by the Include community as an aggregate and harmonized context, but immediately using those data sets, engage the type of questions that were cropping up in the chat as Micah was presenting. Specifically, the unmet need in most contexts is not only pointing and cataloging the sample or aliquot that were used to generate a proteomic profile or an RNA-seq data set, but to support that recursive process of asking the question, if this is true based on the data Joaquin or Micah generated, I want to test this next hypothesis with another data modality or another assay. Is there sample available for that application? Or based on these results that we now understand, are there enough samples to do an additional cohort of discovery by a new modality, long-read sequencing, expanding the proteomic, metabolomic context? And how can we support that iterative cycle of discovery, referencing to the biosamples, and then additional discovery in an integrated way? Like we have been pursuing at the data layer of connecting across portals and resources to allow the intersection of data sets that exist in a cloud-based data fluid environment. As part of this pilot, we have been able to demonstrate the feasibility of connecting two pilot biorepository endpoints, one at the Children's Hospital of Philadelphia and the second at the Cernic Institute. And you'll hear about that next following my presentation. But the really exciting part is that this is just the tip of the iceberg of where the INCLUDE data ecosystem as a whole is heading. And as many of you know, there are several RFAs out right now that begin to describe this emerging landscape that this sets the stage for when you begin to fully connect across biosamples, clinical, and other participant phenotypes, the derived data products, the derived analytic products in a integrated fashion. And more importantly, coming back to Joaquin's set point early on, in ways that is not serial, but co-occurring in real time. And so in the upcoming year, as described by the INCLUDE programmatic setting, there'll be a federated biobanking initiative that takes some of the lessons learned through the pilot efforts that you'll hear about and expands it to create a federated biobank for the INCLUDE program that not only supports prospective sample-based collections, but can interact with existing biosample repositories, and more importantly, can directly interact with the INCLUDE Data Hub and the associated accessibility and use of those samples, associated data sets in an ongoing fashion. And paired with that biosample and biorepository network will be the initiation of really a formalized downstream cohort research site framework where multiple sites will be engaged in a coordinated fashion to collect samples, collect clinical and other features, and integrate those with the ongoing sample setting in ways that's coordinated by a clinical cohort coordinating center. Part of our excitement is that this is an unprecedented infrastructure at scale. And for us, this will require continuous feedback loops from the community partners across every single domain, whether that is the participant community themselves, the researchers, the biobanks, the clinical infrastructure, and then those that have been practicing and supporting research for as long as they've been doing so, and bringing them into that ecosystem in a way that augments their capacity to drive discovery. So you'll hear next from Perrette and JP about this pilot effort to integrate institutional biorepositories into the INCLUDE Data Hub framework, and how that creates a real-time framework of transparent visibility on behalf of a researcher's quest to design a project or design an experiment. But that is in context of what is going to be a much broader and scaled initiative supported by the INCLUDE program in ways that we at the Data Hub are extremely excited to innovate on and co-develop. And like I said, to our knowledge at least, there has never been such a real-time infrastructure that spans all the way from biosample to data generation to information and curation frameworks that derives knowledge and then translational setting as a common and coordinated framework at scale. That setting, as driven across that framework, will be continuously informed by additional data modalities that currently exist and are not yet fully supported, and those that will continue to emerge in upcoming cohorts. And if you review the EXA1 announcement that Wei-Ching mentioned at the beginning of the presentation, you'll notice the call out for what are really a diverse potential of data modalities, data types as generated from derived biosamples. And that is true not just at the biosample layer, but that's true also at other layers of the data ecosystem. And in one specific case that we've been working on hard through a pilot over the past year, this includes imaging-based data workflows. So in the coming year, what you'll see is the full operationalizing of imaging-based workflows as an integrated infrastructure into the Include Data Hub. We honed in initially through the initial period of the project on really supporting these large multi-omic contexts and their provisioning, and are now beginning to intersect these multimodal frameworks, of which imaging is one key component. Over the past year, this required us to really address a complex workflow that can ensure that we can bring in and support the files that are oftentimes generated at the clinical interface, support their harmonization, the identification, and positioning for research use in a unified framework, then process and derive initial derived data products that are easily accessible and utilized by, and can be utilized by the research community. And then importantly, that these output files, once are published into the portal, again, and this is an ongoing experiment, do not create a separate imaging siloed environment that only imaging-based researchers might leverage, but really support bringing that imaging analytic community into proximity and collaboration with the genomics community, the clinical informatics community. And this, again, is a highly unique feature of the Include program. If you look across the NIH landscape and in many other programs, historically, the instinct was to create differential commons, a genomics commons, a proteomics commons, an imaging commons, recognizing that each one of these data modalities requires discrete expertise and a focused community. However, when that happens, you also create silos and missed opportunities for co-discovery and cross-leveraging with the kind of discoveries that Micah highlighted, which is that as you begin to add layers of interpretation across different modalities, you begin to create new structures in that data that support refinement of understanding of the participants' course and can provide tremendous predictive power. To date, imaging, like biosamples, has not yet been a modality in parity to genomics or clinical data. We hope to change that in the coming year based on this pilot that we successfully completed recently, that advances this process of both the integration of clinical and phenotypic data connected to imaging files. And here you see an initial mock-up of the exploration framework of integrating imaging into the portal, but also in ways that support the combining of these multimodal data into a common cloud-enabled workspace environment where the analytic and research communities that span across these multimodal and multi-omic setting can actually iteratively engage in analysis that's empowered from their lens of unmet needs within these cloud-based environment. And you'll hear a lot more of that in the coming year. Between these two efforts of supporting and bringing up into parity biosamples, clinical phenotypic elements, ingesting cohort-based studies, as well as more bespoke and hypothesis-driven research on the Include landscape, and then continuing to support that framework across what is one of the most diverse NIH portfolios of projects, modalities, and resources. I think it provides not only a new way to support the Down syndrome research initiative, but really is an emerging model for how cross-discipline research can occur in ways that drives a precision or personalized translational framework on behalf of those who are participating in research. And for us, that's a really exciting, innovative resource that it has really broad-based impacts beyond even the Down syndrome research setting. With that, I'll turn it over to our two partners, Pierrette Lowe at Sage and JP at St. Justine, who will really walk you through the implementation of the virtual biorepository, in part to highlight the required thoughtfulness that has to take place, even when you deal with biosamples, around how you structure and formalize the collection of biosamples if you hope to create a national and interoperable framework in biosamples that match and are peer to the interoperable molecular data sets. And then really, JP will take you through a real-time query as these features have recently been deployed within the portal. So with that, I'll pause and turn it over to Pierrette. And Pierrette, I think I'll go ahead and advance the slides for you before turning it over to JP.
Thanks. Do we have any questions for Adam first?
Yep, I can pause and take questions based on this. You might have more questions after you see the presentations, which are tightly linked to what I just described, but if not, happy to take questions presently.
Thank you, Adam, for that great background. I'll keep monitoring here the chat for any questions that may arise. Pierrette, all yours.
Okay, thank you. So I'm Pierrette Lowe. I'm a biomedical data manager at Sage BioNetworks, and I handle the data intake and processing for the INCLUDE DCC. So as Adam mentioned, I'm going to give a brief overview of the work that we've done to set up the virtual biorepository, or VBR. And then Jean-Philippe will lead a demo to show you what you can expect as a user. Next slide, please. So the overall vision for the VBR is to allow biobanks that have specimens from participants with Down syndrome to partner with us at the INCLUDE DCC and display their specimen catalogs in the Data Hub. And our goal is to facilitate further research and collaborations by allowing researchers to search for available specimens and then connect with our partner biobanks to request them. And when available, the relevant clinical and sequencing data that is already in the Data Hub will be linked to these specimens, so users can pair this VBR feature with the cohort building and analytics capabilities of the Data Hub. Next slide. So if you are a Data Hub user and you've been analyzing omics data from the Hub, as Adam mentioned, you've already been able to obtain metadata about the associated specimens through the existing biospecimens tab of the data exploration page. But now, if you want to perform further studies on specific samples, so either of the same sample type, or maybe you want to do a different assay on a different sample type from the same participant, you can identify available samples in the Hub and save them as a set to request them from the originating biobank. And then if you've used the Data Hub to assemble a cohort of participants with specific clinical characteristics or co-occurring conditions that you're interested in, you can also identify available samples that way and request them from the biobank. If you're a data contributor and you don't have a biobank, our data model will still allow you to provide metadata about specimens that's focused on their relation to the assay data. But now, if you do have a biobank, you can provide us with a detailed catalog of your specimens that you have available for sharing, and we can display that in the Hub. So it's important to note that the VBR is meant to facilitate a conversation between biobanks and users, but we're not managing the samples themselves or actually facilitating the material transfer agreements. Next slide. So the addition of this VBR required us to expand our existing INCLUDE data model so that we could capture the additional specimen information. And I've shown here on the right the general structure of the new data model. where at the bottom, the most granular level is containers or aliquots of a sample. And then moving up, you have the sample and its parent sample. And then at the top of the hierarchy, there's a collection, which is the material that's actually obtained directly from the participant. So each level is derived from the higher level by laboratory procedures that we are capturing. And we also have information about container storage, volume, concentration, etc. And so many of these things are already represented in the biospecimens tab on the hub, but we've been working on formalizing this model and really defining all the components that we're requesting from a contributor. So the demographic and clinical data are still linked to the participant level, and then the omics and other data files are linked at the sample level. So some of the challenges we faced in building this data model included being able to capture and present sufficient metadata about samples for all of the use cases that I described earlier. So facilitating analyses, allowing users to identify available samples that they're interested in, and then allowing the biobanks to actually find the samples that they need to retrieve. So there are likely to be multiple containers per sample, and also multiple samples of different types for each parent or collection. So for example, you might have plasma samples and whole blood cells or white blood cell samples derived from the same whole blood collection. And so biobanks might be using different lab management systems, and they're likely only tracking sample or aliquot IDs. So as Adam mentioned, the LIMS software isn't necessarily suited to providing information about these other levels like parent samples and collections. But we do need information about all of these levels so that we can link samples that are related to each other. So we have had to work closely with partner biobanks to help them try to create new systems of identifiers for these other levels. And then the final challenge, or well, for now, is that we need to manage all of these identifiers and all of this metadata in a constantly changing catalog of samples that we will need to keep updated on a regular and hopefully frequent basis. Next slide, please. So I'll just show a specific example of the biospecimen data that we obtained from the biobank at the Cernic Institute, which has been providing all of the Human Trisome Project data in the Data Hub. So starting at the top, we have a participant for whom we have demographics, co-occurring conditions, and other clinical data in the Data Hub. And then a collection of whole blood was obtained from this participant by a blood draw. And then a variety of parent samples like plasma, various cell types, etc., were obtained from this whole blood sample by further lab procedures. So I've shown here the example of a parent sample of white blood cells. And then this white blood cell sample was further processed to obtain a sample of white blood cell DNA, which was then aliquoted into multiple containers. And that is what's being tracked in the biobank's freezer system. So additionally, whole genome sequencing was performed on the DNA sample. So in the hub, we have UCF and CRAM files that are linked to this sample. And then the hub will show the sample as available if any of its containers are available. So a user of the Data Hub could arrive at the sample by searching either the participant or the data file tabs. And then since the sample is available, they can request it from the biobank. Next slide, please. So finally, I'll give a brief overview of the request process, which JP will show you next. So we've added 2 new features to the interface of the Data Hub. The first one is the Request Biospecimen button that will take your selected samples and generate a downloadable report that you can use to initiate the request with the biobank. And then we've also added the ability to save sets of requested samples so you can track your request history in your user dashboard. So if you have a biobank and you're interested in partnering with the Include DCC, I'll put our email in the chat. It's info@includedcc.org, and you can reach out there and let us know what you're thinking. So I will let JP introduce himself and give his demo. Thank you.
Thank you, Pierrette. Adam, yeah, awesome. So I'll just share my little screen here. So everyone should see this. So thank you again, Pierrette. Let me just move this little Zoom window here. So thank you, Pierrette, and thank you everyone for joining today's webinar. My name is Jean-Philippe Sibert. I am the product owner / analyst for the Include Data Hub software development team, working under the Saint-Justine Research Center in Montreal. So as Pierrette highlighted and mentioned, I'm going to be mostly touching on two new functionalities for the biospecimen request. So those are two new functionalities, but I wanted to take a little bit of time here to give a brief overview of our current data exploration tool, mostly because it's our exploratory tool for cohort building activities, especially for the newer attendees today. So jumping into the portal here, you'll land on a dashboard, which is more so your personalized, customizable workspace for data navigation and just exploring data in general. We have a few different widgets that facilitate the view of controlled data if you have Air Commons ID and credentials and with dbGaP permission, so the database and phenotype database. And we also have Chromatica projects. This is an API to collaborate with or connect your projects with a cloud-based analytics platform called Chromatica under Valsera's. This is a tool under Valsera's umbrella. So jumping very quickly here, we have, again, the hub for those that don't know, and essentially we have 11 studies for various harmonized data, and we have the count for all of these different studies and the data types here. And before, I just wanted to do a quick touch on the variance page as well. This is a variance exploration page, which we touched on the last webinar, but it's a really cool feature, so I wanted to highlight it very, very quickly. So what this page does, essentially, it enables users to query across millions of annotated germline variants that were essentially taken from Include's participants' genomic files, so they're VCF files. And what we did is that we enriched those VCF files and joined that data with public external databases like DBS and FP, ensemble consequences, ClinVar, various prediction scores, gene to phenotype association. So it's a really great and excellent tool for hypothesis generation. So feel free, take a look. If you haven't done so, I encourage everyone to hop on and take a look at this page. Now, really, for the focus of today's webinar is the data exploration page and specifically the virtual biorepository. So I'll just do a quick refresher about what this page does and how it's useful for researchers and users. Essentially, this is really the cohort building activity tool where it's conducted across 3 different entities: the participant, the biospecimen, and the data files. So for the participant, we have search criteria for clinical annotations with phenotypes within the HPO ontology, diagnoses with the Mondo ontology, various demographics. For the sample type, we have for the biospecimen category, we have various information about the different samples that we have in the portal. So plasma, white blood cells, red blood cells. As Pierrette highlighted, we have collection, sample events and types, age, etc., the availability, the laboratory procedure that was used across the different steps of that hierarchy of sample lineage. And finally, we have the different data files here. So again, spanning a spectrum of different omics that we have in the portal. And so users can use the different search criteria here to look for specific data types, for example, like align reads, gene expression quantifications, or specific methodologies like RNA-seq or WGS. And so here we have also, from the tabular side of things on the right-hand side, we have a summary view, which is a little bit more of a digestible view for charts for our visual folks to get data aggregates. we have the tabular information for the different participant at a row level and their associated metadata. We also have a data files tab where we provide information at a file level, and also we indicate the authorization level depending on the type of data, if it's controlled access, registered tier access, etc. And just a quick highlight, we added a file manifest here, so now we can generate a report showing the file association to participant and biospecimens along with other file metadata. So I think that's going to be useful downstream analyses. And finally, the major focus here is the biospecimens view. So what you're seeing here is essentially what Pierrette was showing in her slide, where you have a sample ID, a parent sample ID, a collection ID, and a container ID. So what this represents, essentially, we have our collection ID, or a collection sample event, which is essentially the extraction of a primary sample type. So that could be blood, whole bloods, a tissue, a saliva, for example. And so what happens is that once, as you showed, those higher level or top level samples could experience certain derivations or processing or treatments or aliquoting. And so what we're really trying to show here is that we're capturing every step of that hierarchy or sample lineage. So let's say you have a peripheral whole blood. Well, that may undergo a certain RBC, red blood cell lysis, to get white blood cells, okay? And that white blood cells would then become a sample ID in our model. But if that that sample for white blood cells, we also did another DNA purification or an extraction with a specific collection kit, or even an RNA extraction / purification, then you would get an RNA sample, which would represent, again, a sample in our model. And so these different samples are also have the aliquots, as she showed. So a sample could have multiple aliquots, and we define that as biologically equivalent aliquots. And this is shown here within our container ID. So what that means is that essentially a sample could have multiple rows, but differing aliquots here. So we capture the volume, etc. So that's great information. And finally, the last thing I want to touch here very quickly is that within a collection ID, you could click here also a hyperlink to get all the samples that fall under that umbrella of that top level. So that peripheral whole blood generated all these different samples: RNA, white blood cells, plasma, etc. And so what that represents is really our first phase of the virtual biorepository. So until now, we've had the metadata about the samples, we've had the sample availability, but we never really had a mechanism to bridge between a user / researcher and the biobanks that have these samples. And so that's the reason why we added this request biospecimen button. And so what that will do is that essentially it's going to generate a report containing the contact information for the biobank, along with specific biospecimen metadata, a detailed list of all the specimens that you select here in the table. And from there, you could do a manual request to the biobank's REDCap form, or if it's just a simple email, then you could send them an email and start that process. So every different biobank will have a different protocol to eventually provision you with those biospecimens. So this is why we added this tool. So where it really gets interesting is, as everyone has been highlighting up to now, is that this is all within this the context of our data exploration and cohort building activities tool. So it's really a combination of, you have a VBR where you're requesting biospecimens, but you could also define very specific search criteria for a clinical background of interest or a specific research question. So with that, let's ask Joaquin, do you have an example of a specific query that's interesting with a specific diagnosis that we could do here? And we'll show an example of how we would do a request biospecimen here.
I have multiple, I always do, including some inspired here by the questions. So let's start with something easy. Can you please find me plasma samples from plasma samples, great, thank you, from females with Down syndrome. And let me add a co-occurring condition and say celiac disease.
Awesome. So we'll go in here in our ontology browser and select celiac disease. So we can see that we have 19 participants. So thank you, Joaquin. So this is really where it gets interesting. We have 19 participants based on the specific query that have all of these that applies to these search filters. And these 19 participants have biospecimens that are within the realm of a sample type that's plasma. So they also have other biospecimens, but now we filtered for plasma. So what would happen is that the user would select all of these results. Let's say they will want all 396 plasma samples or containers. They would click here, request biospecimen, and what you would get is a short description of the action that's taking place. So essentially, you're going to be downloading a manifest and different, a README, essentially guiding you through the biobank-specific protocols to request. We also asked to add a specific biospecimen request name. So here we could add celiac disease for T21 and female, and for plasma samples. And we also get a quick overview. So essentially, if we had selected multiple, if our selection was even broader and it captured multiple biobanks, then you would have a list of 2 different or 3 different biobanks. But in this case, it's only touching 1. So we have 19 participants, and it's important to note here that not all the samples that you have in your selection are going to be available. So here we're really filtering for the available samples here. That are associated to 396 containers or aliquots. So of these 36, we have X amount that have, let's say, 10 or 20 aliquots of the sort. So you could click here, download manifest, and that's going to generate a file. But before jumping into that, I just want to show a few more queries, and then I'll show the manifest. So another interesting thing that you could do here, and if you want to do a little bit more of a comparative analysis, you could duplicate this query. And instead of just having celiac disease, we'll have the opposite. So individuals that haven't been annotated with celiac disease. And so you could also do another similar, a similar request biospecimen here. So let's do one last query here. Still selecting very similar search filters here. So let's do again T21. So try some.
Can I do one based on the questions?
Yeah, sure.
So, the topic of race ethnicity came out from some of our friends in Latin America. So let's say males with trisomy 21, ethnicity Hispanics.
Let's see here. Ethnicity Hispanic. Do we have that? Yeah, we have some.
There we go.
There you go.
Great.
So you'll see here we have the 395 participants for 1,529, and we could see the distribution of sample types here. So there's a myriad of different types of sample types that are available. So it's great. You could select that and request that, and then the biobank will handle that request essentially. Awesome.
An important part, I wonder, Joaquin or JP, just to clarify that, in that biosample view, you're going to get biosamples that may have data already generated, but you may also get biosamples that do not have data generated. And that's, again, something that you can assess by hopping between the views across these cohorts.
Exactly. And we could do a quick example of that. Thank you, Adam. So let's do another query, and let's show that use case where essentially let's build a similar query where you have females with T21, and then again with plasma samples, let's say. And so let's say you want to look at the data files that were generated based on this specific cohort. So the 247 that have plasma. Let's go take a look at what data files they have. So we'll see here that we're spanning really proteomics, metabolomics for protein abundance files that generated, essentially these plasma samples generated protein abundance files and metabolite abundance files. But what happens if you want to expand that research question? And as we've been seeing with Micah's presentation, Adam's presentation, Pierrette, touching on the multi-omic analyses. So what happens if we want to look at essentially gene expression profiles for this specific cohort? And so what you could do is essentially these participants have a bunch of other biospecimens other than this plasma. So as I was showing earlier, we have the collection sample type peripheral whole blood. So you could generate a hypothesis here where, let's say that you could save the participant set here. Let's say with plasma samples. just to be clear. And so what you could do is come back to the participant category. You reuse that set that we just created for those 247 participants, but now we have a lot more biospecimens available. And so you'll see here that all of these samples are available. And essentially, you could do a derivation from the peripheral whole blood to, let's say, get these specific, I'm assuming that, so let's go take a look here at the different data files that are available. And you see here now we have transcriptomics, genomics files, but it was used with the same collection event sample. So what you could do essentially is filter for RNA-seq, for example, if you want to, again, broaden your research question to address some gene expression profiles. And you'll see you have that list of RNA samples. So what happens here is that you have like a multimodal analysis that's already available, where essentially you have a much more complete and comprehensive analysis that could be done across proteomics, through the RNA-seq profiling, and getting that gene expression profiles, and also match the results that we're having here for the proteomics and metabolomics. So it really proves to be a powerful tool. So let's use this last query as the manifest generation here. So let's write RNA-seq. I think it was T21 and female. Let's just say that. So what happens is that it generates the report here as a zip file. So again, showing there's a README and also a CSV format. What you'll get, there you go. What you'll get essentially, so currently, again, as I mentioned, we only had one biobank in this selection, but you would have multiple information, multiple lines for the different studies, essentially. So what you have here is a biobank contact. So that's who the user and researcher is going to communicate with directly. We have the request web link if they have a REDCap form or some sort of form or redirect that facilitates the request. And so we get the detailed information on the biospecimen metadata. So again, this could be tweaked, modified by the user and researcher prior to the request, but it gives a very solid basis to working with the request. So coming back here again, we created this biospecimen request widget, which shows essentially what I just registered as a sample list. So what that means is that users can come back to the request and get the list of samples that they used for the request. Again, for more if they're working with the biobank lab manager, they could refine their search based on a clinical background of interest or the research question. So this could be facilitated with the biobank lab manager. And what we're really trying to do is, so this redirect in that list of samples can also be copied and shared to whoever you're working with. So what we're really trying to do here is, so as you can see, it created a new query. But if I shared that with Joaquin or Adam, then they would also get this new list of biospecimens, which correlates with the request. But what we're really trying to do here is create a workspace where the Include Data Hub, cohort building activities, and biospecimen requests can all be done in one place in collaboration with the biobank to really facilitate that discovery down the line. Because different users, researchers are going to surely use different assays, et cetera. There's going to be new instruments that are going to be available for the different experimental strategies. So that's what we're really trying to empower here. So that's what I wanted to show today. Thank you again to everyone that contributed to the development of this feature, whether that be the biobanks contributing their data, the data and processing teams, of course, the development team that built the features, and all of you for joining the webinar. I think we're going to be closing and asking some opening it for questions and comments now.
Thank you, JP. Yeah.
I just wanted to highlight, one and just touch on what Perrette also pointed out, which is, from the Data Hub perspective, these workflows of biospecimen-based discoveries are really that, right? They're really just to support the discovery that a biospecimen exists and connect it in a meaningful way to existing data sets or cohorts, while still the governance and framework of collaboration or downstream activities are going to be taking place at the biorepository or collaborator level. And so the entire process downstream, which is often the case, includes cohort refinement, assessment, approval, all of those things, still remain under the governance of the biosample curator themselves. Our goal is really to empower that discovery process, support enhanced collaborations, and opportunities to intersect resources that might exist across a differential space. So you can imagine a researcher might have, a grant funding opportunity, an XO1 or a specific hypothesis that can then drive a collaborative interaction with a biobank and engage their biosample request process on their end. So I just wanted to make that clear, that and part of the reason for making that clear is because we understand that this is a heterogeneous space, or as Mike called it, a variegated space. And so there's going to be a continual need for input to us as we do this. And that's a key sort of last message I just want to make sure to send, is that as you engage the portal, as you engage the team here, we really are in a constant phase of getting feedback to improve and learn, from these experiments. So please don't be shy. Please ask questions. Please use the portal-based features to provide feedback and define unmet needs or opportunities for us to improve.
Thank you, Adam. Well said. Huiqing, any parting thoughts or comments?
Yeah, I think, thank you for the whole DCC team for a great presentation. I think it's very exciting to see the, Mecca's presentation using the data, to empower research. Yeah, thanks, everybody, again. One thing I forgot to mention that starting from this year, the XO1 for the omics evaluation, so you can propose all kinds of omics studies. However, for some of them, we are trying to make a rich resource for omics data, all kinds of omics data. However, you can also propose long reads. For long reads, we want you to have a whole genome sequencing for the short reads first. That's the only thing I want to single out. But for we have additional opportunities for omics studies. And you are highly encouraged to use the cloud, the Kavatica platform. If you have any suggestions, we welcome you to share with us. So anything, yeah.
And then maybe just to touch on that briefly, for those of you, many of us are still transitioning from local computational environments to cloud-based resources. And so the DRC has ongoing office hours associated with the use of cloud. So, again, please engage, comment, and, it's a main focus for us to enable cloud-based resources as non-local scalable environments. But we still recognize that this is still an ongoing transition for most of the community.
Yeah, and specifically, please let me know if you have any funding request, if we see any gaps, we'll try to cover them in future.
Very good. Thank you, Huiqing. That's a great note to end. If you need money, ask me. If you need more funding, let me know and we'll look into it. That's great. Very good. Thank you very much to all of our team members here on the DCC for a great set of presentations, and all the attendees from all over the world. Too, I see Aileen from Singapore, Celine from, and I think Anne-Sophie was here from France as well. See colleagues from Mexico, Venezuela, Colombia. Such a wonderful group of colleagues. Until the next one.